Assalamualaikum warahmatullahi wabarakatuh. I am Madam Nazira Zubair. Today we'll take a look at one type of asexual reproduction, which is binary fission. So the learning outcome for today is students should be able to describe what is binary fission. Binary fission is a reproduction strategy or reproduction method in prokaryotes. Example of prokaryotes are bacteria and archaebacteria. The word fission itself refers to splitting or dividing. Fission in biology is division of single entity into two or more parts and the regeneration of those parts to separate entities resembling the original. The object experiencing fission is usually a cell, but the term may also refer to how organisms, bodies, populations, or species split into discrete parts. In the process of binary fission, an organism duplicates its genetic material or replicates its genetic material or DNA and then divides into two parts via cytokinesis, with each new organism receiving one copy of DNA. Alright, next we'll take a look at example of dividing bacterium. These bacteria divide into two cells. Bacteria cells normally divide in exponential way from 1 cell to 2 cells, 4 cells, then 8, 16, and so forth. So there's an exponential increase in the number of cells. Next, we're going to discuss the binary fission using a figure here extracted from Cambridge 11th edition, figure 12.12, page 293. This figure is on bacterial cell division by binary fission. The bacterium E. coli, shown here, has a single circular chromosome. Okay, this figure highlights on the four processes that are very important or vital in the binary fission. And before that, please take note on the origin of replication here. The one that is highlighted in green. This origin of replication is a specific site or specific place on the chromosome where the DNA of the bacterial chromosome begins to replicate. Now we move on to the first process. In the E. coli, the process of cell division is initiated when the DNA of the bacterial chromosome begins to replicate at the origin of replication and producing two origins, as you can see here, two copies of origin. As the chromosome continues to replicate, one origin moves rapidly toward the opposite end of the cell. While the chromosome is replicating, the cell elongates. When replication is complete and the bacterium has reached about twice its initial size, its plasma membrane, this shown at process number 3. So the plasma membrane pinches inward, dividing the parent E. coli cell into two daughter cells. So number 4, the two daughter cells now are produced. So this is the summary of the binary fission that occurs in the E. coli cell. So hopefully that everyone has a clear picture on what is binary fission and how does the binary fission occurs in the bacteria or in E. coli cell. So hope to see you in the next video. See you. Assalamualaikum warahmatullahi wabarakatuh.
Most bacteria reproduce by a process called binary fission. During binary fission, the parental cell divides to form two daughter cells. In the first step of binary fission, the bacterium replicates or copies its chromosome and attaches each identical copy to a separate location on the cell membrane. Unlike eukaryotic cells, bacteria do not need to dissolve a nuclear membrane or assemble a mitotic spindle, making binary fission very quick and efficient. Second, the cell elongates. Since the chromosomes are attached to different locations on the cell membrane, they separate from one another. Third, the bacterium pinches off at the center and forms a septum, a separating wall between the two compartments of the cell. Finally, the cell splits into two new daughter cells. These daughter cells are fully mature bacteria that can grow and, if conditions are right, continue the binary fission process.